the biographical information for Dr. Fredrick Ayuke. He has a very distinguished record working in land management and conservation agriculture. He's worked with CIAT in Nairobi, also at the University of Nairobi, with the Department of Land Resource Management and Agricultural Technology. He has served on several scientific advisory committees and is well published. And it's a delight for me to introduce to you our next speaker. Welcome, Dr. Fredrick Ayuke.
Thank you. Okay. Good afternoon. Yeah, I know it's sometimes very difficult to give a talk, especially after some heavy meal, like we had, of course. But I will give it a try. So, as you have been introduced, that's the much I could also have said. But I want to take you through my talk this afternoon, which will be on biodiversity and productivity under conservation agriculture systems. So I'm presently with the Rwanda Institute for Conservation Agriculture, which is actually just starting. And we are putting infrastructure in place to ensure that we have the first cohort trained the next 3 years so that we have the experiential kind of learning. So when I talk about biodiversity, actually, I will talk about the food security, which, as we know, is that for a growing population, this has been a challenge. And the scenarios that we actually have is that we normally have depleting natural resources and a negative impact of climate variability, and also the cost of inputs, which sometimes becomes unavailable to farmers, as well as the volatile food prices. These ones are actually some of the challenges that farmers do experience. And in addition to some of these challenges, we actually have got indicators, the principles of which we have the soil erosion, soil organic matter decline, and salinization. So this one, all this normally come as a result of tillage-induced soil organic matter decline, or also certain times also structural degradation. So where we actually have got intensive tillage here, we also have got the degradation resulting as a result, and then also insufficient return of organic matter. So presently in Kenya, we have got a problem that quite a number of the soils are already non-responsive, especially to fertilizer application, for reasons that we'll be able to tell as we go about this presentation. So the key thing that has actually been attributed to the non-responsiveness of our soils to fertilizer application is sometimes the insufficient amount of organic matter that would actually cope the soil degradation. So we also have got the insufficient return of the organic materials and also the monocropping. So we think that probably a paradigm shift in the farming practices through elimination of some of the unsustainable practices maybe might be crucial in helping us to gain some of the losses that we've actually made in terms of the soil productivity. So this is actually the point at which I think that the conservation agriculture could be offering some solutions. So when I talk about the conservation agriculture here, we are trying to look at an approach that is actually helping us to maintain an agro-ecosystem so that we actually have got improved productivity, and also we talk about the improved profit and food security. And while we try to do this to improve the food security as well as improving our productivity, we shouldn't be forgetting about our resource base and much more the environment, because these things are all intertwined, and we feel that we need to work in tandem and in close partnership to ensure that the environment is actually not compromised. So when we talk about the conservation agriculture, it's actually characterized by 3 main principles, and we are not limiting ourselves to just the 3. There's also something much more about how do we conserve even parts of the environment. So, but the characterization that we talk about and the principles here largely talk about the minimal or mechanical, minimal soil disturbance, and also permanent soil cover, as well as crop diversification. So all these technologies, these principles, when we combine them, then they actually have got the advantage that they give us opportunity in which we can reduce the cost of production and we increase the yield. At a point we think about our resource as well as the benefit to the environment. So when we look at one of the principles, that is the minimization, especially of degradation here, if we talk about the minimum soil disturbance, that is either through reduced or zero till, it helps us actually to maintain proportion, especially reducing the respiratory gases. We also benefit from organic matter oxidation that is actually moderated when we reduce the tillage, and also we enhance the porosity for water movement and retention. So we also maybe sometimes we limit the exposure of weed to seed such that we enhance the germination. There are quite a number of seeds. Maybe I saw like we had some seed experts here, and they will agree with me that there are certain seeds that will actually take, as long as the soil is infertile, when the soil is actually still fertile and able to support the growth, like nitrogen is still sufficient in the soil, some of these weed seeds do not germinate. For instance, striga. We are told that striga can take as long as 100 years, 50 plus years. It remains in the soil, residence in the soil, as long as the nutrient content is actually high, they will still actually persist. But the moment the nutrient level goes down, like particularly nitrogen, that's the time when they actually come up. So sometimes we think about the minimum tillage here will also be a case example and also a perfect opportunity to think about how do we reduce this. So these ones are some of the examples that we actually have of the reduced tillage or the minimum tillage. We may have a rig line planting here, or maybe sometime mulch tillage, where people just put the mulch on top, it reduces the weed growth, and then they plant in between. Also, we can also think about the planting in basins where we actually have got lots and lots of mulch around, so we don't do the cultivation, and then we can also do the zonal tillage. So the benefits are, as I mentioned, that you are trying to reduce also the weed growth and also disturbance within the soil and also volatilization. So another principle that we also think about in conservation agriculture is the cover, which is actually normally very, very key in our soil conservation aspect. So we think about the permanent soil cover, either can be in the form of organic soil cover in here. We can also think about the cover crop. Early this morning, or maybe mid-morning, we actually had got a talk about the cover crops and also how we can maintain those cover crops. They are actually very much more useful, especially in the context of conservation agriculture. So the benefit of this cover here is that it will actually be protecting our soil against exposure to the effect of maybe sun and also rain. It will also provide a conducive environment for our below biodiversity. The below-ground biodiversity, the macrofauna are actually able to benefit from this because they have a constant supply of food in this regard. Also, the moderation of the microclimate. When we actually have got a cover, then we have the improvement in terms of the microclimate. Then there's also the improvement in the soil aggregation, especially when we have the biological activities and the biodiversity as well as the carbon sequestration. When this, our cover, they rot, they are recycled into the soil, and then we find that carbon is actually sequestered within this. So that's why certain times when we don't have the three components, that is one of them, the biology, and we are destroying the biology because we don't have food for them. That means that we are likely to not to have a functional soil. Probably we speculate that that could be the main reason why we are having non-responsive soil in most of our regions, because we find that we are pumping lots and lots of fertilizer without any organic matter, where even we mine the nutrients, we take away the crop residues as we feed this one to animals, then it becomes a challenge, because anybody starved will not wish to stay where there's no food. So that's actually the same case with organisms. Where there's no food, they move away. And when they move away, that means that there's no structural modification of the soil, there's no movement up and down the soil, then the porosity is also interfered with. So that's why we feel that there's actual linkage between the three properties of the soil. So also the third component that is also very important also in conservation agriculture is also the crop diversification. Apart from it enabling farmers to have a variety or broadening their diet, and also maybe their source of income, I found that sometimes there's also the benefit that the nutrients sometimes that can be deeper in the soil can be drawn up into the upper layer. And also we find that in a diversified kind of system, we are also promoting the diversity in terms of the flora and also the fauna. Because when you have the diversity in terms of the crops here, then you find that you stand the benefit of the environment being modified, and then the organisms are also able to get a broad diet, then they are able to survive in such an environment. So even some of the sequence also help in building the rate of the population pest through the disruption. So where we actually have got the crop rotation here, and we actually have got the pest, then we find that maybe when you rotate this one, then you are breaking the cycle. And we feel that also again in the mid-morning we had a mention about the problem of the fallworm, armyworm. This is actually already, we had the first cohort of students at RICA, the Rwanda Institute for Conservation Agriculture, and among the crops that were actually planting in their first year activities is maize. And it has largely been affected by the armyworms. How I wish that probably had known that we need to use the ash. Maybe we could have thought about that, but we've been trying to use the chemicals. That the damage has actually been very, very great. So despite the wide interest actually in conservation agriculture, and also in many other regions, we found that largely in sub-Saharan Africa, this has been a technology that has not been so much embraced for reasons that we'll be talking about when we'll be mentioning the challenges that we have, especially in Africa. But we feel that maybe we might be talking about the conservation agriculture and the benefit that it has in terms of the soil physical and also the chemical structure, but the biological component has not been so much given a lot of emphasis. So the direction and also we have very little quantification in terms of the biodiversity, how the conservation agriculture relates so much to the soil fauna. So we find that maybe sometimes response of soil organisms to tillage and available crop residues and the cropping practices is actually largely unclear. So that actually introduced me to a study that we conducted in different parts of Kenya. And we actually wanted to find out how conservation agriculture practices actually play a role in the soil productivity, how these associated practices to do with the conservation agriculture, they relate to the soil fauna richness and abundance. So we conducted a study in which we hypothesized that conservation agriculture and the use of the organic and inorganic inputs, they actually will positively influence the soil fauna richness. So this study, we conducted it in 3 different sites. That is in the western part of the country, and then also in the central highlands of Kenya. So this is actually the middle. Actually had got different soil types for these regions. And there were also different agroecological zones, and also with the different soil types and different characteristics. And among the tests that we actually wanted to do was to find out some of the conservation agriculture practices, like the minimum zero tillage versus conventional tillage. Also, what happens with the rotation practices vis-a-vis where we actually added organic residues and where we excluded the organic residues. So in one of the sites, so we actually had got the Embu, which is in the central highlands. We had the continuous conventional tillage maize bean intercrop, and also the soil bean minus and the plus residue. So we are actually looking at some of the practices, because we know that farmers will hardly practice conservation agriculture without residue. Where they actually do the conventional tillage, in most cases they don't apply the organic residue. So we wanted to find out that. And so we actually took some of the practices that farmers themselves do, where there's no any input to represent the farmer's practice. And in most cases, so we wanted to find out also what happens in terms of the rotation versus where we have got the soil practice. This is actually some of the trials that we actually conducted, and we had areas where we were adding the organic residues. So this is the conventional till, which means that we are looking at this one as a farmer's practice, and we have the conventional till where we had the organic residue. Then we also have got the conservation agriculture practices, where we have the maize, soybean rotation, and we have plus residues. And this is actually, because these two trials here were being managed by Kenya Agricultural Livestock Research Institute. Former it was a Kenya Agricultural Research Institute, but then they merged them with all the other organizations. So they merged them and then came up with the Kenya Agricultural Livestock. So they are the ones who are managing this trial here, as well as the lower trial. So the middle one here, so Kakamega and Embu, and then this one also had been managed by CIAT for a very long time. So these trials were established at different periods of time. Since 2010, this lower one is actually also an experiment which I did my PhD way back in 2006 to 2010. And it was actually established in 2003. So it has actually been a long-term experiment. But then the two uppermost are actually mid-term experiments. So this one was established in 2010. By the time we went there, about in 2015, 2016, so it was just about 6 years. So the measurements that we actually took was to measure the soil macrofauna, that is the below-ground biodiversity. So we were trying to use the soil macrofauna, those organisms that are above 2 millimeters, to represent the macrofauna. And then we also took measurements about the mesofauna. So these ones are the intermediate between the microflora, the microfauna, as well as the macrofauna. The larger components that constitute earthworms and termites, those ones are the macrofauna. And then the mesofauna, we have the springtails and also the collembola. So those ones were largely constituting the mesofauna. So what we did is that we measured monoliths, soil monoliths, measuring 25 square centimeters, and we went 30 centimeters deep, either using shovel, and also in some cases we were using the hose. So at each observation, what we did is that we measured, we took one monolith per plot, and because we actually had got different replications and also different trials and the components, so we were also able to extend much of this. So this is what is actually representative of what we actually did during the time when we were actually measuring the soil fauna. Then after we have obtained the monolith, then we took this one to the base, just about where we're doing the work. Then we actually sorted out the macrofauna, and we stored this macrofauna in bottles. So on the other hand, we also took measurement for the mesofauna. That is the next category in terms of the body size. And we did this one at the same time when we were actually taking soil sample for the macrofauna. And we measured the metal cores, that is about 10 centimeters in diameter. But then we were also going up to 30 centimeters. So we were taking these measurements in two depths, that is 0 to 15 and also 15 to 30. And then once we had obtained the soil samples, then we took this one to the lab and then used the equipment here. This is the Balise Tolgren apparatus, where we are actually able to subject the soil sample to a gradient of heat, adjusting this one occasionally. So we wanted to ensure that we subject them to some heat here in this soil with some light here. Then we get the soil, the samples put in here, and then we collect our macrofauna down here. So when we adjust our apparatus here, moving it up and down, so it becomes much hotter down here, and then the mesofauna, they move, and then we collect them down in alcohol there. So that is actually how we did the extraction of the mesofauna. And then The fauna groups that we actually collected, we took them to the lab. First of all, like the macrofauna, especially the earthworms, we put them, first of all, we fix them in 4% formaldehyde. And we kill them, first of all, in alcohol, ethanol, and then we fix them in formaldehyde. This is actually to ensure that we don't lose them after some time. Because normally, for earthworms, they secrete some fluid when you put them in alcohol, and then they dilute it, then they can start rotting. So that's actually why we were using the formaldehyde. And then we took this one to the lab, that is in IPBES, that is in CIAT, and then we did the identification of some of these species. So we subjected our data that we got to statistical analysis using the GenStat. And of course, it was actually procedural that we tested for normality in the data, and where possible, we actually did the transformation. So the data that actually, I will not go so much in the details about the much information and about the too much data that we actually took, but I'll just take some of the ones that I feel are much more critical to our talk today. So our significance was actually determined at 5% level. So this is actually part of the results that we actually got. This was actually in Embu. But then what happens is that some of the differences that we observed were largely at the top surface of the soils for all the sites. We didn't find any difference, especially in terms of the site for all the regions. That is below 15 centimeter to 30, we didn't find any difference. But then this one also reinforces the fact that below the ground there, unless maybe conditions are actually not very good, the organisms largely operates at the top. Maybe they will go down when the conditions are not actually very, very much conducive, that they maybe either the temperatures are actually very high and they need a cooler temperature. That's when they actually move down. So that explains why our differences were actually largely recorded. This was actually for macrofauna. So for Embu, and also, okay, sorry for that, please. So for Embu, so if you look at what happens here, is that we actually had got the topsoil. So this is actually, you can look at the conventional agriculture. This is actually what we talk about, the conventional tillage here. Even with the residues here, and then we find that there's actually the soil maize here plus residues, and also the intercropping here. So the performance in conventional tillage here, with or without the residues, actually performing much poorer than we actually had got that. Then you look at the soil bin. This is actually soil bins, where we only had soil bins, organic residue returned into the soil. Again, the performance wasn't that much. So again, this one will be reinforcing what happens is that, do we have enough of these residues resident in the soil? The answer is actually yes and no. Certain times you find that quite a number of these organic residues, they disappear very fast. And especially we realize that this one's actually happening where we actually had got the bin residues here. But where we actually had got the soil maize, it was taking quite some time to decompose, and then the environment is a little bit much modified, and also where we actually had got the conventional tillage. So we are finding that where we had got the maize bin intercrop here, there was actually a little bit of some performance in terms of the diversity. But then in Kakamega, we didn't actually observe any difference among the treatments that we actually had. But nevertheless, you can actually see that conventional till was still relatively higher than this, although not significant. And then you look at in the Nyabeda site, which is actually a very long-term trial. The farmers practice, this is where we represent the conventional till without any input whatsoever, which is actually what they do. It was actually the poorest in terms of the performance. But then you found that even the conventional till where we had got the maize soybean rotation here, plus residues, it was actually not so much difference with the other conventional tillage practices. So we didn't actually find any treatment effect on the macrofauna, especially at lower depths. And also we didn't find any treatment for the mesofauna diversity for all depths. So that means that there was actually selective kind of response with regard to some of the practices that we actually had, and especially given that this was across the different agroecological regions, then probably something could have been happening. And then this was actually also the mesofauna abundance, that is in terms of the numbers. And you can actually see that the farmers' practice as well as the convention till is actually, they are at the lowest here. So where we actually had got the maize bean intercrop here with residues, there was actually some good performance. But then we found that except for this top layer in Kakamega area, there was not so much difference for all the other sites. So what do we say? Something that we actually had hypothesized is that we'll be able to get some more information with regard that macrofauna will actually be higher under conservation where we have the residues than where we don't have the residues, which is actually true. But then we found that the medium to long-term addition of residues also is enhancing the fauna in terms of richness and abundance. But then also we found that there was actually increase. The conservation agriculture is increasing our taxonomic richness as well as abundance when we compare this one with the conventional till. But under the maize bean intercropping, the performance is actually much higher than where you actually have got the soil beans. And this was actually much more, especially where we have the maize bean intercrop. So I will actually say that adoption of conservation agriculture would actually be very, very much important for us because it's actually a way in which we can have the soil fauna enhanced. But something that is actually remarkable that we can actually note is that we have numerous challenges with regard to the smallholder farmers adopting some of these conservation agriculture technologies or maybe practices, because sometimes you find that they may rarely apply all the three principles at the same time. A farmer may apply the residues. He doesn't feel the need to practice maybe minimum tillage or maybe crop rotation, or he practices crop rotation, but at the end of it, remove the crop residues from the farm, feed this one to the animals, then it actually becomes a challenge. But then maybe it will actually be much more interesting if we can have the simultaneous interactions, especially of these components, where it can actually be studied. So all is not lost, but we must appreciate that we actually have got lots and lots of challenges with regard to the conservation agriculture practices. In as much as we may want to try and very fast, then we find that we still have got lots and lots of challenges about our adoption of this. For instance, we actually have got the chief most is a competition of the crop residues between the conservation agriculture practices and also the uses that we might want to use this one for. For instance, in many parts, I believe, and we know, there are quite a number of regions that we feed the organic residues to the animals. And part of which we are talking about the nutrient mining and reduction in organic matter in many parts, for instance, in Kenya, is actually coming as a result of somebody sometimes is actually buying the green maize from the farmer. He pays for everything, gets the maize, he cuts everything. So he harvests all the maize stalk, including the stover. He carries everything away, so that he will go and get the cob, probably to roast or maybe to cook, and then the rest are being fed to the animals. And then in other cultures also we have situations where we also use the crop residues as wood fuel. That is actually to augment the fuel that we actually have. And certain times also farmers burn this one. So in that case, we find that the competition for this residue will actually become enormous, such that what is actually left on farm is very little. It can actually be non-sustainable. And then we find that maybe sometimes there might be a little bit lack of knowledge about the potential of conservation agriculture, especially among the agriculturists and also extensionists. And we need to think about how do we strengthen the knowledge and information sharing mechanism so that we actually highlight some of the benefits and also some of the challenges. Because as scientists, we actually have to really acknowledge that we need to share the information, and we also have got challenges with regard to some of these practices. Where we have the practices and we have the challenges, we have to admit that really we have a challenge, because we cannot actually always assume that farmers are ignorant. Farmers themselves also have indigenous knowledge that they can also share with us. And then sometimes also the availability, especially of the skilled and scientific manpower, and how do we overcome the bias and the mindset about tillage. We may think that tillage is actually a problem about bring about the weeds. Tillage is actually a problem that maybe it can also bring about the pest. So how do you overcome that bias? Maybe the mindset that tillage is actually bad. And maybe how do you also overcome the fact that conservation tillage, maybe minimum tillage, can also be a good practice. So we may need to think so much about some of these practices, and how do we actually come up with a policy framework and strategies to promote this. But then sometimes in one of the experiments that we also had got some work done in one of the sites, which is Nyabeda in western part of Kenya, we came to realize that this is an experiment that has actually been conducted over a very long time. But when we conducted a yield analysis, we found that there was no yield difference between the conservation agriculture per se, as well as the conventional agriculture. Actually, relatively conventional tillage practices were even doing much better. But when I went in to conduct some of the studies and also the biology about that experiment, then we realized that one thing is that I was finding more of the organisms nesting outside the field and very few of them in the plot. But then one thing which was also observed is that the termites particularly were taking the organic residues from the field, they take them to the nest outside. And the biology of those species was they don't like any disturbance. So they would rather they feed safely somewhere away from that your tillage practice, but they get their food. So during the night, they go to the field, they go and get the residue that you have actually put there, they take them to their nest. And then so we didn't find any benefit. There was actually no benefit whatsoever from the conservation tillage practices, especially where the organic residues were actually being used. So that is something that we also need to think about, because if we don't have the three practices intertwined together, then it's actually a sorry state that the biology will actually be functioning here. We don't have the infiltration because the organic residues have been taken out of the field, then the chemistry also is affected. So then in that case, you find that the conservation agriculture might not be so much beneficial. That's why we really need to think about how do we actually work in tandem here and share what we actually have and some of the challenges that we can be talking about. So this study was actually supported by SIMLESA, that's Sustainable Land and Extension Services, that is under CIAT, and CIMIT. And we actually were integrated especially to the CALRO staff who facilitated this study, and also IITA. So with that, I wish to stop. Thank you.
Let's give thanks first.